Hi, this is Dr. Launen, and this is the first of two online lectures or two online videos that together compose a lecture explaining or introducing the genomics focus of the Vibrio vulnificus project happening in my lab here at Keene State College. In the previous lecture, which introduced you to the biology of Vibrio vulnificus, I made a case for why it is important to study Vibrio vulnificus in New Hampshire, specifically in the Great Bay Estuary. And the 5 sets of information or pieces of information that contribute to that case are restated here. I also showed you this slide in the previous lecture which depicts our sampling locations. So this in color is the Great Bay Estuary of New Hampshire. And it's our major coastal area in New Hampshire, and it's the location where oysters are collected and also where people spend a lot of recreational time utilizing waters. So this represents a place where Vibrio vulnificus is found. It's certainly located and well established in the Great Bay Estuary. It's abundant in our oysters there, and we are harvesting our oysters for consumption, and that's a growing market in New Hampshire as elsewhere in the United States. The 2 major sampling areas where our oysters or where our Vibrio vulnificus are derived are this Oyster River location. It's a contaminated area subject to contamination from wastewater treatment plants and so forth. We don't harvest oysters there for consumption, but there are a lot of naturally occurring oysters there, and so it's worth monitoring Vibrio vulnificus in that area. And then we have a lot of Vibrio vulnificus from water, sediment, and oysters collected around Nanny Island. And that is a location where there are commercial oyster aquaculture enterprises. We also have a few from the Adams Point area. So this is an estuarine environment, very salty, relatively shallow water compared to a marine system. There's been a Vibrio monitoring program well established since 2007, and that's the source of the isolates that we are getting. That Vibrio monitoring program is run by Dr. Stephen Jones at the University of New Hampshire, and he's a collaborator on this project. We receive samples from his laboratory at regular intervals, and then we characterize those Vibrio vulnificus here at Keene State College. And what we've noticed in recent years is that we seem to be recovering more Vibrio vulnificus per sampling event. That suggests a shift in the populations in the Great Bay Estuary because the total numbers aren't increasing. So if the total numbers are the same, but yet we recover more per sampling event, more specific individual isolates, that means that the isolates themselves might be more culturable, easier to grow in the lab, and that suggests they may be changing. Our project goals are to genetically characterize Vibrio vulnificus from the Great Bay Estuary, working with isolates obtained since 2007, and to determine how they respond to environmental conditions, particularly how it affects their growth rates. Our hypotheses are the following. We hypothesize that the Vibrio vulnificus strains in the Great Bay Estuary are non-pathogenic and are most genetically similar to environmental strains found in other regions. Why that hypothesis? Well, because to our knowledge there has been no actual infection due to a Vibrio vulnificus contracted from the Great Bay Estuary region. We also hypothesize that the Vibrio vulnificus strains in the Great Bay Estuary are adapting to environmental change, specifically temperature and salinity in the region. So they're adapting to the increasing seawater temperatures that we're seeing here as elsewhere on the planet. This is the workflow for the project. So isolates of Vibrio vulnificus, and here are some growing on a Petri plate, are obtained from the University of New Hampshire. We confirm, what we do is we look at them on a plate and make sure each colony looks the same. That's an indicator of purity for each particular strain that is being cultured. We then extract genomic or gDNA from the isolates, and we use PCR using a PCR reaction that amplifies a species-specific marker gene, the VVH gene, found only in Vibrio vulnificus. So a band on the PCR means you've got Vibrio vulnificus. A lack of band indicates that you do not. That tells us that we've got genomic DNA and it's good quality and it's actually from a Vibrio vulnificus. We then sequence it, and we don't do this ourselves, but we submit it to the Hubbard Genome Center at the University of New Hampshire, and they conduct Illumina next generation sequencing, and it's 250 base pair paired end reads as explained previously. And then that data gets funneled into two pipelines at this point. The first pipeline is a pipeline where we do gene-specific analysis, and I'll talk more about that in a moment. So we pick a few genes, and we focus on those genes to understand our isolates. The other pipeline, which is the part that you're working on as part of your class project, if you're listening to this as a Bio 311 student, is the whole genome analysis or comparative genomic approach. And I'll talk about that more in a moment as well. So one of the things that you have to do if you're going to culture Vibrio vulnificus in your laboratory is you have to respect the organism and you have to follow the appropriate rules and protocols. So priority 1 in our lab environment is to establish what are called Biosafety Level 2 areas and protocols and training methodologies. So because this is a potentially dangerous pathogen, we have to be sure that we handle the organism very carefully. This is Sarah Sanders, and that's me, Dr. Launen, and Sarah's working in what's called a biosafety cabinet. So this protects her from Vibrio vulnificus, and it also protects Vibrio vulnificus from her, or more specifically the microbes that are on Sarah and all of us. And so we wear double gloves and lab coats, and we wear eye protection whenever we're handling the organism. And we have very specific containment and sterilization policies for all materials that contact the organism because we don't ever want to have an infection in our lab. This is probably more precautionary than we need to be because the isolates that we have are environmental. And as I said before, there's never been an infection due to isolates obtained from the Great Bay Estuary. So probably what we have is relatively harmless, but you can never be too sure. And because Vibrio vulnificus is classified as a BSL-2 organism, that's how we handle it. And this also gives students really great training in how to handle pathogenic microbes. That's useful to them in their future career goals and work. So what we do is we obtain isolates that are from the University of New Hampshire. They would actually go out into the field and collect samples of oysters, water, or sediment, and then they take those samples and they do what's called a most probable number analysis. And this is a process where you make serial dilutions. So this is diluted 100 times, this is where a sample is diluted 1,000 times, and here it's diluted 10,000 times. And you look at the tubes for growth, and you use a scoring matrix that allows you to say, okay, there are, 3 Vibrio vulnificus most probable number units per gram of sample in this particular sample. So this allows us to get the levels of Vibrio vulnificus found in a sample. Now from there, you streak the positive tubes onto a kind of auger called chrome auger, and it's got a color indicator, and so it will show you whether or not you have Vibrio vulnificus or whether the Vibrio is something else. And it'll allow you to separate strains one from another based on color and the colony morphology. Once we've got them purified, we subculture onto tryptic soy auger so that we get high levels of growth, and to verify that the colony morphology is the same, which indicates purity in the strain. And then from there, we grow them up in a broth called HI broth, and that's what we use to extract DNA to do PCR to determine and confirm that what we're handling is Vibrio vulnificus. And we also make freezer cultures at this point so that the strains are archived, and that's a really important step because you can't publish work about strains that aren't archived. Because if they're not archived, you can't go back and repeat the experiments, and that's part of the scientific method. So on that chrome auger that I mentioned a while ago, we don't usually draw letters, but this is just drawn to show us what color Vibrio vulnificus looks like on chrome auger. Vibrio parahemolyticus looks like this. It's got the purple color. So this allows us to differentiate between different kinds of Vibrio, which can be tricky in our lab, but can be really hard to make sure we don't have any Vibrio parahemolyticus mixed in with our Vibrio vulnificus. And we're not interested in our lab in studying VP. And this is Kylie, a student in my lab group, and she's doing PCR. So bands using the VVH PCR primers show us that you do indeed have Vibrio vulnificus present. You should see no bands in the negative controls, and that's indeed what she's seeing. And the positive controls are strains that we obtained from a collaborator from the University of Florida that we know for certain is Vibrio vulnificus, and these are positive bands from 3 New Hampshire isolates.